Welcome. In this video, we're going to discuss the answers to the B2 revision worksheet, as well as some of the questions that are likely to potentially come up on an exam. There is a link down below in the description to the full set of answers for this worksheet if you'd like to go back and check your own work. And of course, if you have any questions on this, please post those in the comments below. We'll be happy to answer those. It begins with talking about diffusion. Just remember that this is a passive movement of particles and it's from higher to lower concentration, and these are two regions that we're talking about. It's a passive movement, which means that it does not require energy, but it will be affected by a number of different things, for example, temperature, membrane, distance, etc. In terms of osmosis, it's very similar to diffusion, but it is involving water that's moving from the high water potential to low water potential, and water potential is essentially the inverse of concentration. So where there are lots of particles, that means that water is going to want to move into that area so that those particles can become diluted if the particles are not able to move out. When you look at carrier proteins, and we've got another one of my spectacular drawings here, you need to remember that they are still proteins. They're usually specific to their substrate. Here's a kind of better drawing here. So what we've got happening is the substrate binding to the active site, and then ATP or energy being used to change the shape of that carrier protein. The substrate is then going to be moved into the cell, and that is against the concentration gradient, and so therefore it requires energy for that process to happen. Remember that when you're looking at these stem cell diagrams, you are looking at differences in terms of the structure and function of those cells, embryonic stem cells being those at the very start, because they can form a number of different cell types, literally any cell type in the body. As stem cells mature and as cells mature, then you might end up having them specialize a little bit more. At number 10, with surface area to volume ratios, be very careful that you get these the right way around. So surface area to volume. That means the surface area needs to come first. That's often a mistake we see. At question 15, the experiment with the potometer, make sure you remember about how that experiment needs to be laid out. Things like ensuring there's a bubble in the tube, ensuring that the reservoir is there to do resets and things like that. That will really help you to be able to describe the experiment in detail on a test.